Good evening, ladies and gentlemen. Can I welcome you to this evening's inaugural lecture, and to those of you who are visitors from outside the university, welcome to Keele University. It's good to see a very diverse audience here this evening, because I know that amongst the attendees we have representatives of quite a number of personal and professional and academic groups with whom Lindsay has been associated. I know we have quite a number of members of Lindsay's family. We certainly have excellent representation from the medical school and from the North Staffordshire health economy, or the Staffordshire health economy more broadly speaking, but also representatives from a number of national groups with which Lindsay has had very close association. So a great welcome to you all, because I'm very much of the view that inaugural lectures are about reaching out to a diverse group of audiences to demonstrate the expertise that the new professor has. And I think that's both a demonstration of that expertise, but also a chance to show some of the thinking, some of the ideas, some of the research and innovation which is going on in a research-led university like Keele. Lindsay, as most of you will know, is now Professor of Applied Medical Sciences here at Keele, but is also Director of Academic Undergraduate Studies in the School of Medicine. Lindsay had a very distinguished educational career of his own as an Oxford undergraduate and DPhil student, and then entered the world of academic research before coming to Keele in 2002. And Lindsay came to Keele at the start of the development of our medical school here to really provide the leadership around the non-clinical components of curriculum in that new medical school. Previously, he'd been Vice Dean of Undergraduate Medicine and Head of the Department of Biochemistry and Immunology at St George's University of London. And that expertise, both in his research and his education, has led to him becoming a Fellow of the Higher Education Academy, and indeed to being a very distinguished academic in his specialist research field. His research over the years has focused on biochemistry and biophysics, particularly in relation to cellular membranes, and that's research using optical and other physical methods to monitor and explain the structure and function of those membranes. And some of his discoveries now inform some of the classic textbook explanations for the transport and storage of various neurotransmitters. That research was a very distinctive feature of Lindsay's profile before he came to Keele, but the reputation and the impact of that research has been sustained right through until the current day, when I think Lindsay is still very much recognised as one of the leading developers within his specialist field of research. In tonight's lecture, Lindsay is going to focus on research, teaching, and the dialogue between those two elements, which lie at the very heart, very core of what a university is. Lindsay's lecture is going to tread a path through his own experience of all three elements, as he's seen them as a student, as a researcher, as a teacher, and as a leader. Asking questions such as, how does great research inform the best teaching? How are the very different cultures of humanities and the natural sciences balanced in programmes like Lindsay's particular interest, medicine, that require equal measures of both the natural sciences, the physical sciences, and the humanities? So I know that Lindsay is going to explore how medical schools can use the interplay between art and the sciences to produce graduates who are able to make informed judgments on the basis of often imperfect evidence. So can I therefore formally welcome Lindsay Bashford, Professor of Applied Medical Sciences, and invite him to present his inaugural lecture, Culture, Curiosity and Conversation, Three Pillars of the Academy. Lindsay.
Thank you very much, Vice-Chancellor. always slightly embarrassing to hear all these things said about your career at the beginning of these lectures, and also to see, it's really nice to see all you people here who've all various strands of my life, and I hope there's something for you to take home after we've finished. And I particularly, when it comes to thanks, it's always difficult to know who to thank most, but I'm glad to see that Rhiannon and Sarah sitting together there at the back. Without them, none of this, of all the things that have happened today, could have properly happened. So it's really nice to see them here and able to do that. And thank you both very much who've been supporting me in that activity. So what do we really want to do with the family, friends, church? How do you deal with an inaugural lecture? Well, journey is right. So the Vice-Chancellor said from student. Well, I think first pictures I've got is a little bit of a personal journey, and then you'll try and see how these three themes of curiosity, culture, and conversation fits together. So if you look at that now, these, who is this person? Well, this is me, aged about 6. This is the most special photograph that my wife possesses, given to her by my mum. Sadly, mum can't be here, and nor can dad for that matter, but I'm sure they're very pleased that I'm here. It's really good to have such parents who have encouraged everything that I've done. This was given to Barbara, my dear wife, on the strict instructions that it was not to be lost. And it actually took a bit of finding to be used for this lecture. But there it is. So that's me, age 6, probably just left Cambridge then, which was where I was born. So that's a 50s picture. That's a 50s picture. This is a 60s, late 60s, early 70s picture to show that I had grown up. And you can see the hair was a bit longer then. That's not the longest haired picture I've got of me as a student. But there are some, but it's certainly a lot darker than it was. But you can see that I was on my way. Then I think here I'm an undergraduate student at Oxford University. And then just to see how the journey develops, here I am as a research assistant in the Johnson Research Foundation in the University of Pennsylvania. That's my desk. That had daylight in it. You'll see that's significant later on. And there's this delightful person beside me. That's Barbara. And actually at that time, that's only a few months before my daughter Susan was born. So she's in that picture as well. But you can see there we are. And she'd come to work in the same institute where I was, because it's quite tricky for a primary school teacher to get a job in the United States. And so, in fact, she had to go back to university for 3 years, and we weren't sure we'd be there that long. So the boss said, well, what can you do? He said, well, I drew all the diagrams in Lindsay's thesis, and within a few weeks, she was the artist for the Johnson Research Foundation and remained that for the rest of the time we were there. So that was fantastic. There's employment is very flexible in the United States, not always as easy to do it in the United Kingdom. But anyway, that was a great time, but we didn't stay in Philadelphia for 2 main reasons. I didn't really want to be in a country that didn't play cricket. That's a very important factor. And we were neither of us really sure that we wanted our daughter to be an all-American girl. Right, so those are 2 things that meant, so the family was quite strong. So we came back to London when there was a job in St George's. And here I am in the lab at St George's, shared with Charles, who's down there in the front row, and he'll recognise when that was taken. I think that's sometime around in the 80s. It might be a bit later, but it was meant to be, this was the 70s, this was the 80s. All I can say for certain is that, isn't it interesting that I was even then doing experiments in a biological laboratory wearing a tie? And I still continue to do that. But this is definitely my own blood, because you were not allowed to use anybody else's blood for experiments. So we could have our blood do it and look at the cells. We'll talk about the cells in a minute. That laboratory I shared with Charles, and we shared a lot together, and I owe him a lot for being able to give me the opportunity to study and work at St George's. My actual office in St George's was possibly the gloomiest office you could ever imagine. And I actually loved it. It had no windows, it had no paint, it had these breeze block walls all the way around, all 4. There was a little window there. Had to use the lights all the time. You'll see later why I quite like dark rooms, but no windows for 20 years. I still managed to smile then. My hair might have been going a bit grey. The architect assured me that this breeze block, specially beautifully pointed, you can see there, that's called modern marble, he said, in this new block. I wasn't completely convinced about that. And then when you come to Keele, here I am here. This is in the 2000s. And so I'd come to Keele. That's a picture taken of me. I think probably, you'll see, I was doing problem-based learning tutorial when that was taken. So that was my journey to get to Keele to be a member of staff in the medical school there. And that sort of doesn't really tell you much about the culture, the curiosity and the conversation. So let's think about curiosity. And I was thinking, how can we have an image that everybody will grasp that it's all about curiosity? And I thought this from my family album was an absolutely splendid one. So here is my granddaughter, Evie. She's in the garden, and she's extremely interested in these white berries, which she's able to eat. And I thought that was just an absolute lovely picture of curiosity. And I hope my curiosity, when I was that 6-year-old boy that you saw earlier, I wanted to know everything about everything. I didn't think about science, I didn't think about medicine, I didn't think about art or music. If there's something that I didn't know, I wanted to know a bit more about it. And that was an important thing to do. And so here, I think this is Evie showing her concentration with her grandpa in the garden. I thought that's a really nice picture to get into the notion of curiosity. And this might be botany, might be biology, might be science, but one of the themes that I like to develop is that I was born when C.P. Snow wrote about the two cultures, which was arts and humanities, and and sciences. And Keele was growing up as a university, as an integrated university where arts and sciences were meant to be done together. So that was all in the background before coming together to make a medical school where I think you need to have both the science basis and the humanities basis to get properly to understand the human condition and to make doctors as good as they can be. And so that's a little bit where I'm going to go. I just thought this was a lovely picture about curiosity. And so if we pick up the sort of strands of work, we'll start probably with the science, which is where my own curiosity comes from. How do things work? Why do they work? Why doesn't that piece of information, why doesn't that quite fit in what we thought was the proper story? There's something else going on. And so it's that natural inquisitiveness, which I think in science is essentially optimistic. I'm not so clear-cut about humanities. The curiosity can have both pessimistic and optimistic things, but I think generally in science, the expectation is that you'll ask a question to which you'll get an answer, and that everyone will be better informed at the end of it. And so there's that optimistic strand in it in science, which I think is a positive. So where are we going to go from there? He said, not looking at his notes and wondering, yes. So my sense now, I think the lights were going to dim here, but you can probably see pretty well that, as the Vice-Chancellor said, I've been really interested in the cell membrane. Now, this cell on the left, as you look at it, that's a liver cell, grown by my friend Pete Knox, and allowed me to look at it in his very fancy microscope. And around the edge of the cell is the cell membrane that you'll see I'll talk to you about more in a moment. And inside, there are all these little wriggly bits and black dots and worms. And so cells are actually full of stuff. And you think, well, how important are those? We've got 10 to the 13, 10 to the 14 cells in our body, all of them surrounded by a membrane, all working well together. So all our nerve, all our muscle, all our blood, all of that stuff is all made up of individual cells, and they've all got to work together in harmony. And in order for them to work, they've got to have stuff that goes in and stuff that comes out. And all of that has to go across a membrane. And so all my research career has been thinking about the membranes. And so this is a bit of optical microscopy, very fancy optical microscopy of a liver cell, and you can see its structure. On the right is a picture I took, and I'm so proud of it. I've used it ever since I took it, and it's just fantastic. It's the sort of thing you can do on a Friday afternoon, this, and so when nobody else is using the very fancy microscope that's sitting there in your department. You think, well, I wonder what these cells look like if you put this dye in them. And that's exactly what this is. So this is now something called a 3T3 cell. That's a cell line that's been used for a very long time to look at cell studies. Both these cells are grown in tissue culture, so you can study them very easily, and you can look at their properties, and you can actually do these wonderful microscopes. And what I've given them here is a dye, a positively charged dye, which you shine blue light on it, and it glows green. Now, that's a really important, that's called fluorescence, and for those of you who are physical scientists will understand that light comes in at a higher energy, that's the blue end. It loses a bit of its energy before it's emitted from the molecule, comes out green. And so we shone a blue light, and you take a picture with the green on, so you can't see the light that's exciting it because it doesn't go through your filters. So you get this beautiful cell popping out. And the point of this dye is it crosses the membrane very easily and goes wherever there's a lot of negative potential. So these wriggly worms, which are the same as these ones here, and they're called mitochondria, they're very strongly negative, inside negative, so the dye piles up inside them. And I'm quite interested in the membrane that makes that strong potential. And around the edge of the cell, you can see the green here. That's where the cell surface membrane is, and you can actually tell the cell from the outside because the cell membrane is also has the inside negative, and so the dye goes into the cell and accumulates in the cytoplasm, and it takes it up from the outside. So the outside is dark, the cell surface just has this greenish hue, but because these mitochondria are very strongly negative, nearly all the dye goes inside. And the message from that is that you can use the dyes, shine them at the cells, and see changes, and that can make you understand what's going on inside the cells. And so that takes me, these are quite late on. I took these, I was at St George's in Charles's department when we did that work. But before that, I'd already done some work with fluorescence to see, could you explore what was going on? In fact, in a little vesicle like one of these black things in there, but it wasn't from the liver, it was from nerve cells. And it resulted in these papers, which were done when I was a graduate student in the 1970s. And George Rada, who is not here tonight, I remain eternally grateful for him because he said I could come and do a PhD in his lab, pretty much provided I passed my final exam, because I had been doing a project with him, had a very serious accident when I was knocked off my bicycle, could do no work for the last 6 months of my final year as an undergraduate, took my exams, got a degree, not as good as I might have liked, but he said, don't worry, Lindsay, you can come and do your PhD in my lab. So I'm eternally grateful for him for giving me that opportunity. And within a couple of years, we got these papers, which are about Chroma granules. Now, not many people will have heard of Chroma granules. They're the little storage vesicles, surrounded by a membrane, just like the cell membrane, but more tightly curved, and they store the hormones adrenaline and noradrenaline, which everyone will have heard about. Those are the things that are making my heart beat at a good rate now, a little bit faster than usual, a little bit of drive there from the adrenaline to keep it going during a lecture. And they're stored in that, and they're released when they're needed. And so they have to be taken up into the granules, stored there, and released when necessary. And what we discovered in these using dyes was the mechanism by which that happened. And so this is what we actually found. So this is the key experiment here. This is in black and white, a beautiful Barbara diagram from my PhD thesis long ago that got her that job later on. And so if you imagine here are our chromaffin granules just sitting in a tube and we're shining a light at them. We haven't, there's no dye added, so there's no signal, so it's right down at naught. We add the dye, and you see it goes up, signal goes up, so there's a little bit of fluorescence. We give them energy, you can't read that, but it says ATP there. That's giving the transporters energy to work, and lo and behold, look at that, the fluorescence went up fantastically. And so we thought, this is a negatively charged dye, fluorescence is going up, that must mean we're making the inside positive. And that indeed was what it happens. And then we thought, if it's in, what could be moving? And the answer was it had to be a positively charged something, and it turned out to be the hydrogen ion, H+, that was moving. So if you added to the membrane something that made them leak hydrogen ions, they all came out and the potential went away. So that's a real-time experiment. We thought, I wonder if that's what's driving the noradrenaline and the adrenaline onto the inside. So we did the experiment here. So we made these vesicles, we took all the stuff outside of them, we reconstituted them empty, gave them energy, and lo and behold, they took adrenaline up just beautifully in the presence of ATP. If you gave this thing that made the membrane leak to protons, virtually none went in. So that told us that we've got this mechanism by which a pump here, this is my model, would push protons across the membrane, making it positive inside, and those protons would come out. As they came out, they'd take the catecholamine in, and that would give you catecholamine stored on this side. So that was my model 1976. To give you an idea that people were still thinking about what we did then, here in 2004, somebody else's work, working on similar systems, not in exactly the same place, and they produced this lovely diagram. You know, we've come into color and all of that sort of thing and fancy drawings, but here ATPA is pumping the protons in. That was there in my simple model. There it is, much more structurally involved, and we do know that the structure of the protein is like that with all these things inside the membrane and this structure's outside. We know that's the truth now. Here's the transporter that takes the catecholamine in. This one is called DA, that's for dopamine rather than noradrenaline, but the mechanism is similar. And that's how it works, and actually you know that for every dopamine that came in, 2 protons came out. That means you get a really good, strong accumulation on that side. So that's work done in a simple experiment as a graduate student when I was young, learning about publication, learning about competition, all those sorts of things. And you look back and think, you discovered something really quite exciting, and you published it in actually quite, not top-line journals, but respected journals, rather than anywhere else. And part of that was, and it's part of the scientific culture, is that we thought we discovered something really important, and we wanted to be the first to be published. And that is a general motivating force of science. It's good in many ways, and it encourages you to do good experiments, to write them up and get them published. It's bad from my other point of view of the conversation, because it means often you don't fully reveal in your conversations with your fellow scientists what you've been doing just in case you get scooped. Now, whether that applies in the humanities as well, I'm looking at Bill there, and I suspect exactly the same sort of thing. It's that academic competition, wanting priority, wanting to be the best, wanting to be the first. That's part and parcel of academic life, and you have to learn to live with it. So it has good things, but there are some downsides to it as well. So that's one research story. A second research story, this is Charles is here, and so here is C.A. Pasternak, his department. We worked together for many years, and we were very interested in a cell line that we were using to study how toxins damage them, and we'll talk a bit more about that. And I just did this experiment because I thought, well, perhaps when the toxins damage the cell, they'll affect its membrane potential. And so I had, by now, a slightly different dye to measure the membrane potential. And we did this classic experiment down here. So we gave the dye and we got a signal, and we are now measuring its absorption rather than its fluorescence. And we thought, all right, that's all right. I wonder what happens when we depolarize the cell. Cells normally are negative inside and positive outside. Normally you depolarize them by giving them potassium, and that depolarizes them. And so we did that, and we thought, well, that ought to do it, because normally they're charged up by potassium leaking out. Now we'll make it go in. And absolutely nothing happened. And we were baffled, completely baffled, I think we'll say about it. And so then we had in our armory another drug that made the membrane permeable to potassium. And if you did that, they depolarized, just as you'd expect. So let's do the experiment the other way around. Let's add the potassium channel first. And then that made the membranes actually hyperpolarize them quite a lot, and then you depolarize them. So I thought, this looks like a membrane that's making its potential without using the classical methods of cells, which is to pump the ions, get gradients, and then they leak down. I tried to present that to the Physiology Society in St. George's. So this is an anecdote to go with that now. And they looked at it and said, that can't possibly be true. They said, everybody knows that the equations for membrane potential tell you that potassium depolarizes cell. This can't be true. It must be a mistake in your method, right? And actually, because we can't see what the mistake is, and you're quite persuasive, and you've done all sorts of experiments that makes it look as though your method is telling the truth, we'll send you off to Cambridge so that you have to give your talk again to people who are better qualified to pass judgment. Charles thought that was great for me. He said, Lindsay, that will be just fantastic. You'll go and do that. So then we went off to Cambridge, and we were in this Biochemical Society meeting, and we were there all morning, and our talk was last. And he kept nudging me as we went in. He said, oh, you know, that's Huxley's just come in. He said, he's come in to listen to your talk. He said, that's Glyn. He's chairing your talk. These are all the Nobel laureates who'd written those equations that said this was impossible. But fortunately, they're broad-minded, open-minded, not prejudiced, and they accepted the data and said, yeah, you can publish it, and that's all right. Then, probably 15 years later, in the lab, we got a different technique available where you could measure the potential directly, just as the physiologists wanted us to. And there it is, minus 30, minus 40. At rest, you inhibit the pump, and it goes down to 0. So if we'd done that experiment when we did that one, everyone would have believed us, but you had to, you know, so it tells you in science that you do need to get over a bit of the talk, you know, the pressure that your experiments need to show what they're meant to be showing. So that was an interesting story. It took us a little time, I think, to try and persuade people that that was a proper way to think about things. And so then I'm only doing 3 bits of research, and so if one is the sort of 70s, this is the 80s, the next one is the 90s, and this again was following up. We wanted, and this is a truly esoteric slide, we wanted to know, could we find the holes that we thought that bacterial toxin or that virus fusing with the cell membrane or that plant protein toxin, or we knew they made holes in membranes because we knew they made stuff leak out that shouldn't leak out, and they let nasty stuff in that shouldn't go in. So the membrane definitely had holes in it. And nobody really knew how big they were, and we thought, could we find them in a microscope and see how big they were? And this was my idea. And so I went to a meeting at the Royal Society where somebody discussed force microscopes. Now, I have to, provided you can see me, the way a force microscope works, it's harder to see there, easier to see, is that it scans across a surface like this, backwards and forwards. You imagine the back, it comes to an object, it goes up over the object, and then it goes down again. So you've got a very fine tip going up. So I've been over my glass, it carries on, it's flat, it comes up over the glass. And so whenever it does, tip has to come back. Shine a laser onto the back of my hand, or the tip of the microscope, and you can know exactly the position of the tip at every point of that scan, like a TV scan raster. And you do that. Our only difference was that we didn't use something to touch it, we used this glass pipette, and we measured the current coming through the glass pipette. And the answer is when you got really close, there was no current because it sealed off, pulled it off a little bit, you could do it. And then it could scan over the surface as much as you like, and you could keep it so that the current leaking through, that's what this curve down here is just kept constant. And then it would just scan over, always keeping the same distance off the surface. So what will happen if you look at cells? And so now we've got some more beautiful pictures, maybe slightly dimmer lights for this as well. So now these are the sorts of pictures that my friends who took electron microscopy, where you had to fix the cell, stain it, remove all the stuff, put heavy metals onto it and things like that, they looked exactly like that, except these cells are now growing just like the other ones were, in a dish, perfectly alive. There are 3 different sorts of cell. The melanocytes are there, the cells that give you the skin pigment, dark skin pigment. CaCO2 cells, they're a cancer cell line from a human colonic cancer cell, and cardiac myocytes are the muscle cells in hearts. And you can see that when you use this, it just creeping over them like that, you've got this wonderful sensitive feeling of the cell surface. And actually, Yuri, who did these experiments for us, you could see them waving, you could see them, and you could understand what they were doing. And so we had this beautiful picture of the cell surface. And we had a chance of finding the holes. And actually, in the myocytes, which have those ion channels that allow the potassium to go in, the calcium to go in, he was actually able to find those with the microscope. And again, it was a bit of another of those things that the physicists who I explained this to, they just laughed. I mean, if I thought the physiologists at Cambridge were sceptical, the physicists in Durham were outrageous. They said, you've got absolutely no chance of seeing the holes that you want to see with your glass pipette that's 10 times their diameter. It just can't be done. And actually, you could find them, but you couldn't really get at their structure. And so it turned out that the sensitivity of our microscope was not the diameter of the pipette tip, but how close it went to the surface. So there was a bit of right on both sides. And as soon as they saw the pictures, they said, actually, you're onto something really good there. So if you like to think, that's my bit on my curiosity, the things that I've wanted to do to understand how cells work, how they fit together. And you can see that probably not every doctor has to know in quite that level of detail all those things working. But some of them who are wanting to really get to grips with what's going on in illnesses will want to know that about the things that they're particularly keen on. So when they come to their education, you've got to have the ability to be able to go right to the cutting edge, as well as at that level of generality where there's understanding, so that you can inform patients properly about treatments. So then I wanted to go to the conversation bit. Now, that's really hard, again, to find pictures about conversation. So I've got sort of 3 categories of conversation. This is my father-in-law, now 93, and this is his elder brother, now 96 or 97. And they're having a conversation in a pub in Yorkshire a couple of summers ago, and I took the picture. And I thought, isn't that just a lovely picture of them? And I thought, from the point of view of conversation of people who've got different backgrounds, these are 2 people with very similar backgrounds, and they talked and talked and talked. They don't meet up very often, and they just talk to each other, and you could sense their common culture, their common experience. So they had a very deep understanding of each other, and that even though neither of them can hear almost anything, right, because they're both, you can see they've both got full hearing aids. They love talking about their hearing aids and how to make them work, but actually they understood each other really well, and that's part of the shared experience, part of the common culture, so they knew what each other was going to say, a lifetime of family reactions. And I think you can't capture that in a picture, but I think it is a nice picture of two people having a conversation. This is people of like minds having a conversation. We can go forward a bit now. This is a picture that we use in the medical school to illustrate how different elements of learning are required. So these are medical students here, and this is Lauren Brooks, one of our health sociologists, and this is a learning experience where for medical students at this stage of their career, they're normally pretty skeptical about how useful health sociology will be in their learning, when what they really want to know is how do those cells work, what's gone wrong in the pathology of those illnesses. And they don't always necessarily get that feel that actually they need to think both about that and how to relate to the person and to get the whole holistic picture. So this is, I think, a conversation where there's slightly more challenge on the two sides of the equation. So there's an enthusiastic, very knowledgeable, very capable health sociologist here, Lauren, speaking to slightly more skeptical students who need to pick up the useful things that they can pick up and from a whole range of their teachers. And it is my thesis that to make a really great medical school, you've got to have the ability to have the conversations of people of all sorts of different backgrounds and understandings, because that's the only way that the doctors will learn about the human condition. And so they've got to do it. So that was my sort of contrast to the so there's the formal like-mindedness of Ken and his brother. Here is where you've got to cross-cultural, got to have a cross-cultural conversation. And so we have to arrange that to happen for our medical students. And then my third conversation, I thought this is another lovely picture from the family album. And here is my son-in-law, can't be with us tonight, and two of my grandchildren, and a hen. All right, and so they're clearly having a conversation about looking after hens, making happy hens, learning about how hens work, what they eat, but it's this circle of trust and understanding and acceptance and excitement and trying to understand what's going on. And I thought that's a very nice, that gives that sort of family view of conversations and the shared meaning and shared values, which are so important for our doctors to be, that they have good, solid values that they can share with all sorts and conditions of people. And I think that's a very interesting thing. And so you've got to have conversations in your schools that enable like minds with like minds, counter minds with counter minds, and across generations and across understanding and across all disciplines. Whatever you call chicken studies, I don't know. But the idea is that you've got to be able to have that flexibility and adaptability. And I think that's really important to encourage our students to do that. And so having had all those pictures, I now have a diagram just of text written by Sir David Watson that I read in the Times Higher whenever it was, 6 years ago now, 7 years ago now, well, 6 and a bit years ago. And I think that encapsulates what I think about what education is in higher education and in medical schools with which I am most familiar. It is that conversation. It's the conversation between the staff and the students. Most of what I've learned as an undergraduate student and then as a researcher has been refocused and recharged and reorganized by the questions that the students ask you in tutorials. And they say, well, why is this? And you scratch your head and say, I actually have no idea why that is, but I can't not give them an answer, so I need to think of an answer and tell them. And then they look at me as adoringly and think, that's the truth. I said, no, don't believe that a word. That's just the answer I've made up from what I know. You've got to go away and check out whether it's right or wrong. And so then you've started to engage in the conversation between you and the students. And it's the same with the researchers in the lab. It's that wherever you go, if you don't have that dialogue, that conversation where you have shared ground and you can understand what you're talking about, then that's what you've got to do. And so all the time, I think, while we've been building up our medical school at Keele, it's this goal of being able to have that conversation. I happen to know we've got an orthopedic surgeon in the audience, we've got another surgeon in the audience, we've got general practitioners, we've got sociologists, psychologists, scientists. They've all got to be able to have those conversations with each other of proper respect and understanding. Now that, of course, doesn't make a medical school. That's a bit of an informal structure for a medical school, so you need a bit more guidelines about that. And so how do we make that work? Well, to make a medical school work, you have to satisfy the General Medical Council. There's no two ways about it. If they don't say your degree's all right, you can't graduate doctors with a degree that allows them to practice medicine. And I remember it very well, not in 2009, they caused an uproar in 1992 or 1993 when they published a document called Tomorrow's Doctors that essentially said that most of you guys in medical schools are teaching medicine in a very old-fashioned way. We don't think you should be doing that, and we are encouraging you to move to a different style. We're now in 2013, and virtually, but not entirely, all medical schools now teach in that new style. So it's taken a long time for that to happen. And they laid that out properly in documents that this is the third edition, Tomorrow's Doctors. This is what, when they graduate from your medical school, they've got to fulfill these requirements. And the requirements are they have to be doctors who are scholars and scientists. I love that. That's great, isn't it? That's right up my street. That's what I'm at Keele for, to make sure that they're scholars and scientists. Doctors are practitioners. That's all my clinical colleagues. They're there to make sure they do their clinical practice as well as they can. And then doctors professional, they've got to have those values to share in the health service, to have core values of compassion and care and putting the patient first, all of that. They've got to learn how to do all those things. And in fact, Tomorrow's Doctors is a bit more detailed than that. I think there are 137 outcomes that we have to sign off that the doctors have to have when they leave, and we're pretty confident that that's what they do. So how can you go about that? And so here I know my Peter Coventry, another good friend, colleague at the medical school, can't be here tonight. Fancy having a daughter's 18th birthday the same day that I'm giving my lecture. But there it is. So he's there. And he introduced me to this slide, which I think is really great. Now, you'll notice all my slides so far have had this Keele graduating excellent clinicians. Andy Hassel is in the audience somewhere. I can't see where he is. And that was he drove us to this thinking, what can we have as a motto, a thing that we can aspire to so that we know that Keele is going to be good. We know that they're going to be clinical when they come out and they're going to be excellent. So he came up with graduating excellent clinicians. So that's right at the center of this bubble diagram. Excellent. How are we going to get there? So this is how do you create a school of medicine, which is no different from creating any other sort of school in academic life that's going to be effective. And so I think these 5 things, so that's a great curriculum there, excellent opportunities to practice, students being given a lot of time to learn for themselves, having the proper professional cultural background, and having good facilities. And actually, as you'll see, going through Keele, we've been able to actually put a little tick in every one of these boxes, and so that's what the last few minutes of this lecture will be about. So if you think about an effective learning, so you want a proper curriculum. We designed our curriculum from the back, so we decided we wanted those outcomes. How do you get there? And we decided that we would have what everybody calls a spiral curriculum. I have illustrated that with this nice helix here, all right? So that you're properly mathematically qualified. That's a helix, not a spiral. And so we have a helical curriculum. That's what most spiral staircases are, helices, not spirals. So just to show that one can go in for the details there. And the idea of the helix is that you go round and round, you go through the material that they need to learn for their medicine, and you keep coming back to the same things, but every time you get to them the next time, you're in a different place, and so you see them from a different perspective, and so the new perspective adds to the old, and when you get to the top, you've fulfilled the learning outcomes of tomorrow's doctors, you've managed all the complexity, and you're ready to go off and practice. So that's the idea and so we do that. We designed the curriculum explicitly. Here are the 3 themes: the doctor as scholar and scientist, doctor as a practitioner, doctor as professional. They run through the whole curriculum. They're not really straight arrows like that. They wriggle around, and sometimes they're thicker, sometimes they're thinner. But by the time you get to the end, they've all been delivered. And then the actual learning opportunities are in blocks and units as you go through year 1 to year 5. It takes a time. So that's a schematic. I like to think when we've finished, what is a doctor, our graduate, going to be like? Now, I have a very particular image, which I've always had, that this curriculum, essentially, you might like to think each of these nice coloured steps on the helix is a coloured brick. And so the student, every learning opportunity they have, they collect another coloured brick. And they're all different colours. And what can you make with that? You can make a mosaic. So I thought, where will I get a picture of a mosaic? And lo and behold, thanks to this Christina Daniel and Google and other such things, here is a early Roman doctor, Saint Pantaleon, in Hosios Lukos Monastery in Greece, Holy Luke. Fantastic. And he's a mosaic, right? And so that's exactly what I was meaning about. They have made the mosaic bricks, make St. Pantaleon. I think each of our students who graduates will have their own bricks, but they will make the picture that's them, that's going to be the surgeon, going to be the general practitioner, going to be the psychiatrist, going to be the lab scientist. They've made that picture into that, and I think that's a really good thing. The other nice thing about this mosaic is a lot of those bricks are gold. Right? And so I think that gives us a good idea of the quality of our learning opportunities. We're giving them golden opportunities to learn well and to do well. So I think that's just a message. So again, it's hard to know what sort of output you want. We want a doctor fit for every action in the health service, so that we've given them a kit to make them available, to make their own mosaic. So then we can come to extensive clinical experience. What do we mean by that? 100 weeks of placements. Actually, to be good at anything, you've got to practice. And then when you're good at it, you've got to continue practicing it. And if you want to keep on doing it, you've got to practice it again. And that's what the cricket told me. You couldn't ever score runs. You might have scored a century in your last innings in 2012, but if you didn't go to the nets when you started in 2013, you were normally out for a low score. Practice is always needed. So we needed to give our students loads of practice. That's what it's for. And these are the places, and we've got fantastic places for them to practice. This is the medical school building on the Kiel campus. This is the Clinical Education Centre on the hospital campus. This, I believe, is the entrance to UHNS, the emergency entrance. This is the Learning Centre in Shrewsbury, where our students spend time. This is the Learning Centre in Stafford, where the students learn. And this is a community practice, where the students learn. So those are the physical places where our students go and learn. And they practice in all those different places so that they get different experiences of the health service, and they're able to put those together to make a good mosaic. Student-centered learning, so we've got to give them opportunities to learn for themselves. Emphasis on feedback and student engagement. So we do everything right in the medical school. The pictures I've got here are just ones that in the David Weatherall building. So you've got this is in our anatomy resource room, where I'm not quite sure what's going on with the dry bone and the fully alive leg, but clearly something important is going on there to identify where that bone should be in the real person. Here is a problem-based learning tutorial with me looking suspiciously like I did in that other photograph there, so I suspect comes from the same thing. Students learning together. This is a student paying a lot of attention to my words in the lecture theatre, or perhaps somebody else's words in the lecture theatre. And here they are doing a practical on themselves, exercise to see how much oxygen they can consume under stress. So we give them all those excellent facilities to learn and practice and to develop the skills they need to become as doctors. Now, feedback. Now, there's a lot in the university. Feedback. The university is all about feedback. The better feedback we give to the students, the more they will learn. They'll get better degrees, or in our case, they'll graduate more excellent clinicians than they might otherwise have been. And they'll give us a good score in the National Student Survey, and so more students will come to Kiel. All of those are really good things to do, but I'm not actually going to talk to you about any of that sort of feedback, because sometimes what's forgotten in feedback in the conversation is that the students who you get to know really well say nice things and some not so nice things about you in the feedback they give you as a teacher. So these are from a couple of my PBL groups, 2010, I can't remember when that one was, who they gave me those at the end of 12 weeks when we'd been together. I really liked Mr. Cheerful. I thought that's a very nice thing. And then they sent this. Interestingly, on the cover, they didn't put a picture of me on it. When they got into the calendar itself, somehow I had joined the group and was one of their pictures. What an interesting thought that was. But they tell you all sorts of interesting things, and that you normally would miss otherwise. And actually, that's the most useful feedback they can give you. Sometimes the feedback is a bit delayed. Now, about 10 years ago, I was talking to someone who was a very distinguished general practitioner who'd been one of my students 15 years before that. And she said, yes, she said, I remember your lecture on vision. Of course, I swelled with pride, remembering a lecture 15 years ago. Isn't that fantastic? I said, yes, what do you remember about it? And she said, well, what was really interesting is that you talked about your son. She said, I remember you saying that your son had asked you why he couldn't see colours by moonlight when he could by sunlight. Now, that's a very deep and difficult question to answer and very interesting. I thought, that's not exactly the explanation. I thought she would tell me something I'd learned about how vision works and rods and cones. But no, she said, isn't it interesting that someone should have a son who would ask a question like that and expect to get an answer? Right? Two things. And she obviously, so it was the human side, and completely different from what you'd expect you'd said in your lecture, a completely different message. And so I think, that's feedback that you need to understand, and sometimes you have to take it a bit on the chin. So we're nearly there. Promotion of professional, socially responsible, globally aware attitudes. Keele's very keen on this. We want our students to be world citizens. We want them to interact with the local community. They're going to be doctors. They're going to be wherever people are unwell, and that's everywhere. So they need to be able to cope with all those sorts of conditions. And we give quite a lot of opportunity for that. And actually, the organisers for some of this are here. And so we thought, how can we demonstrate in a nice, easy, visually attractive way all the organisations with which our students work? And here they all are as their logos. And they're all local organisations, and our students have placements with them in year 2 when they go and work with them for 24 hours, 8 days, and they work with them in year 5 when they do projects to make the community a better place. So if that's not really helping our students to be socially aware and embedded in the local community, I can't think of a better way of doing it. This nice little logo at the bottom here, that's our cluster project logo. That's they work together, 4 or 5 students in their final year, in the group of local practices where they're doing their training, to develop a project, normally with one of these people, to make their community a better place. So I think we're really, really on the ball there at Keele to make a really good curriculum to make socially aware students. International awareness is another thing that we want to do, and we give them the opportunity to go away in year 5 for an 8 weeks abroad. And to make sure that they do something interesting, we give them a prize for the best photo that they bring back from their elective study. And these are the prize winners from the years that you can see in there. And so our students, they were there for 8 weeks. So this is Africa, this is India, Nepal. I'm not actually sure where the other ones are, but immediately you get the idea. Students have been out there, they're working in international environments, that's developing their ability to be global citizens. Not every student goes abroad, so it's not something that they all do, but it is an opportunity that we give them in order to become globally aware as well as locally aware of social issues. Finally, we come to the excellent facilities. You saw the great buildings, and I thought, how can we have another image about great buildings? Well, we can have what goes on inside the buildings. So I asked Mike to give me an image of good things going on inside buildings. And so here are our students. Here's one of our staff, who I can see in the lecture theatre, giving a lecture. Probably that student was paying much more attention to him than would be to me. Here are students working with something called SimMan, which is a model patient where you can do all sorts of things, and so they can learn a lot of clinical skills. Here's someone doing a laboratory activity, and here's someone looking at a histology. So wherever we go, our students can have these excellent facilities and excellent learning opportunities. So that's what it is. How do you do that? Of course, behind that, and lots of them are here, are all my colleagues who have been fantastic to work with, fantastic to help. We've had great heads of school with Sue Dillian, Richard Hayes and Val Wass. We've had all sorts of people who've really made it go. Rhiannon and Zara, I've already talked about, who support us to make it happen. They're the people who make it really work. And the goal and the end is to graduate excellent clinicians. So here are our excellent clinicians graduating. These are our first cohort in 2012. And I like it because the students are all at the top of this slide, clearly the most important place to be. And here are all the staff, slightly more colourful, in the front. If you're really eagle-eyed and you look at me on the stage and then, can I see Lindsay in there? And the answer is, you can't. Like McCavity, I'm not there. This picture was taken before the graduation ceremony. And I was on the train from London, where I'd been doing an external examiner job at another medical school, on my way back to be at the graduation ceremony, which I definitely was at. And the other thing because the pictures are taken in Keele Hall and everybody has to look up at the photographer, all these people at the front and the students all looking up very optimistically, very ambitiously, a really good thing. And finally, the other thing I like about this row is that within that group of staff, there are members of staff who once upon a time were my students in another medical school, and that's always a sense of immense pride that they've come and developed and grown and doing that. And so that's a really good thing. So that's where I think I want to draw to an end. I think I've got one more slide which is just words, and that's to try and draw those themes together. I'm not going to read it out to you, you're going to read it, and I'm just going to say thank you for being such an attentive audience, and I'm sure I shall have a chance to answer some questions in a minute. So thank you all very much indeed.